Hello, Triden and Nagar, and today we're going to be solving May-June 2020 Paper 1 Variant 1. And before we start, I'd highly appreciate it if you would take a moment and subscribe to this channel and like this video if this channel offers you any value. Before we start, we are offering free Topic 1 notes. All you have to do is fill in the form in the description box down below. We are also selling Topic 2 notes, which include all the tips, tricks of Paper 1 and common misconceptions with worked examples too. Upon purchase, you can ask us unlimited number of questions about the topic or past paper questions for free, and you are able to exclusively contact us at any time, which is a new and unique service. We also have another monthly service, which you can ask us unlimited number of questions and past paper questions for the full AS Biology syllabus only for $15 per month. Question number 1. The photomicrograph shows the ultrastructure of part of a cell. Which statement about the type of cell shown in the photomicrograph is correct? Now, this is clearly a plant cell because we can see the cell walls and the chloroplast, because here we could see the grana made of thylakoids, which look like stacks inside the chloroplast. So this is clearly a chloroplast. Now let's see the suggestions that we have here. A, it is a plant cell because it has both chloroplasts and a nucleus. Now this question was a very common misconception because people thought that this structure here was the nucleus. This is incorrect. This cell surprisingly does not have a nucleus, because if you zoomed in further, you would also see the grana and the thylakoids, which means that this is not a nucleus, however it is a chloroplast. Therefore, A is incorrect, and many candidates place the answer as A. B, it is a plant cell because it has chloroplasts. This is correct. We can only see the chloroplasts visible. Therefore, the answer is going to be B. Question number 2. Which structures are found in typical eukaryotic cells? 1. 70S ribosomes. This is correct because 70S ribosomes are found in mitochondria and chloroplasts in eukaryotic cells. 80S ribosomes. This is correct. They are found in the cytoplasm of the eukaryotic cell. 3. Linear DNA, aka chromosomes. This is correct. They are found inside the nucleus. And 4. Circular DNA. As we all know, circular DNA is found in both chloroplasts and mitochondria. Therefore, this is correct, and the answer is going to be A. Question number 3. A student examined a slide of human blood with a light microscope and made a careful drawing of the different cell types. The student used an eyepiece graticule so that the relative sizes of the different cell types were drawn accurately. In the drawing, red blood cells were 7 mm in diameter and so on. What is the linear magnification of the drawing? Now, this was also a very common misconception, but as we all know, and you should know this and memorize it, that red blood cells are actually 7 micrometers in diameter, and this is the actual size. So here we have a standard that we can use to calculate the magnification, and it is a red blood cell. Now, to calculate the magnification, the equation is I am. Therefore, we have to divide the image size, which is 7 millimeters, by the actual size. Now, first what we must do is convert 7 millimeters into micrometers. We do this by multiplying by 1,000. So the image size is going to be 7,000 micrometers. Now, 7,000 divided by 7 gives you 1,000, and the answer is going to be D. Number 4. The diagram shows a granulated slide with divisions of 0.1 millimeters viewed using an eyepiece graticule. Now, let's first start with this part. Let's label them. So this is the granulated slide, and this scale divided into 100 divisions is the eyepiece graticule. Very important to be able to distinguish them in a past paper question. Now here it says that the divisions of the slide is going to be 0.1 millimeters. Now we're trying to convert it to micrometers. We do this by multiplying it by 1,000. This gives us 100 micrometers. So each division gives us 100 micrometers on the slide. Now we are trying to find the length of one division of the eyepiece graticule. To do this, as we can see, that per 100 divisions on the eyepiece graticule, there are 200 micrometers. Now what we must do is we divide 200 micrometers by 100. This gives us, so we divide 200 micrometers by the 100 divisions to calculate the size of one division. This gives us 2 micrometers per division. Now, once we've got this clear, let's continue with our question. Pollen grains were grown in a sugar solution and viewed using the eyepiece graticule. Diagram 1 shows the pollen grains at the start. Diagram 2 shows the pollen grains after 4 hours. Now, 4 hours is incredibly important. Now, what is the growth rate of the pollen tubes? Now, here we have micrometers or millimeters per hour. Now, let's use this one as reference. Now we could see that it has grown approximately 10. And we know that each division up here on the eyepiece graticule is equal to 2 micrometers. Then, if this covers 10 divisions, then what we must do is multiply 10 divisions by 2 micrometers. This would give us 20 micrometers. But this is per 4 hours, because here it says the pollen grains after 4 hours. Now, here we're trying to get the growth rate of the pollen tubes per hour. Then what we must do is we divide 20 micrometers by 4 to calculate the length for 1 hour, and this would give you 5 micrometers per hour. Question number 5. The table shows a comparison between 2 features of a typical eukaryotic cell and a typical prokaryotic cell. Which row shows the correct comparison between these cells? Now, let's start with the presence of centromeres. As we all know that eukaryotic cells definitely do have centromeres. And what is a centromere? It's what holds these sister chromatids, the 2 sister chromatids together in a linear chromosome. And as we all know that inside the nucleus of a eukaryotic cell, we have linear chromosomes, which look like this, so they do contain centromeres. Now, for prokaryotic cells, we know that they have circular DNA, not the normal linear DNA that we have. Therefore, a normal circular DNA would not have centromeres. Therefore, this is correct. Now, presence of telomeres. Now, telomeres are only present at the ends of the chromosomes of linear chromosomes. Therefore, they are only present in eukaryotic cells, and they are not present in prokaryotic cells, as we said, because it is circular DNA. Therefore, the correct answer is going to be B. Question number 6. The flow diagram shows the results of a number of tests on a solution of biochemicals. Which substances are present in the solution? Now, Benedict's test, blue, and Benedict's test used to test for the presence of reducing sugars. And a positive test, it would turn from blue to brick red. Now, because it stayed blue, this means it's a negative test and reducing sugars are not present. Now, non-reducing sugar test, the solution turned yellow. Therefore, there was a partial change in color. Now, how is a non-reducing sugar test different to a normal Benedict's test reducing sugar test? Now, first of all, that in a non-reducing sugar situation, first we have to add hydrochloric acid in order to be able to hydrolyze the glycosidic bond of the disaccharide. That's the only difference. Now, because here we have seen a color change, this means that it is a positive test for non-reducing sugars. Now, for iodine test, it turned from almost a brown color to blue-black. Therefore, it is a positive test for starch. And Biuret test, which turned purple, this is a positive test for proteins. Therefore, non-reducing, starch and proteins are present, and the answer is going to be D. The reason for this is that sucrose is a reducing sugar, starch, as we mentioned here, and catalase. Catalase is an enzyme and it is made up of proteins. Number 7, maltose and sucrose are disaccharides. Maltose is formed from the 2 molecules of glucose, while sucrose is formed from fructose and glucose, which row shows the molecular formulae of the 2 disaccharides. So as we all know, the formula of a normal glucose is C6H12O6. Now, it's a disaccharide, which means that 2 of these have combined together. And as we all know, in a condensation reaction, a molecule of water is removed. So what we must do is multiply all these values by 2, which would give us C12, H24, and O12, and then subtract 2 hydrogens and 1 oxygen from the water molecule that was released in the condensation reaction. That's an easy way to remember the formula. Now, it's going to give us C12, H22, and O11. Therefore, the answer is going to be A. Question number 8. The diagram shows short sections of some common polysaccharides and modified polysaccharides. Which row correctly matches the numbered diagrams to the descriptions of the polysaccharides? Now for F here, it says it's composed of beta-glucose monomers and 1,4 glycosidic bonds. As we all know, beta-glucose monomers, the hydroxyl group is above the ring. Therefore here we're looking at only 1 and 2 in this case. For G, it's composed of alpha-glucose monomers with 1,4 glycosidic bond. As we all know, alpha-glucose monomers, the hydroxyl group is below the ring. Now then it could only be 3. And for H, is composed of N-acetylglucosamine monomers with beta 1 to 4 glycosidic bonds. So here we're also looking at 1 to 2, but here because a nitrogen is mentioned here, the only diagram that shows nitrogen presence is going to be 1. Then H is definitely going to be 1, and then F is going to be 2. Therefore, the answer is going to be B. Which molecules contain at least 2 double bonds? Now, let's start with one unsaturated fatty acids. And here's asking at least 2 double bonds. So, we know that all acids have a carboxylic acid group. And a carboxylic acid group has a double bond present. Therefore, there it contains 1, and because we know it's unsaturated fatty acid, then it must have a double bond between the hydrocarbon tail. Therefore, there's going to be at least a double bond in the unsaturated fatty acids. So here we have at least 2. Therefore, this is correct. For 2, collagen and hemoglobin 3. Now we know that collagen and hemoglobin are proteins made of amino acids. So let's draw the structure of an amino acid. The R is the R group or the side chain. Here's the hydrogen atom, and here is the carboxyl group. Now, here in an amino acid, we could clearly see a double bond in the carboxyl group. Therefore, all proteins must have at least 2 double bonds. So 2 and 3 is correct, and the answer is going to be A. And the diagram shows a protein molecule. 2 long polypeptides form alpha helices for much of their length, and these twist together into a fiber. At one end, each of these polypeptides coils into a globular head. 2 short polypeptides bind to each globular head. What describes the protein structure of this molecule? Now here it says that 2 long polypeptides, and we all know that quaternary structure is the combining of 2 or more polypeptide chains by specific bonds. There are 4 bonds, by the way. Therefore, because here it says that 2 long polypeptides, and here we could see the long and the short combining, therefore it must be quaternary. It cannot be anything else. Therefore, the answer is going to be A. Question number 11. Which types of bond will keep a folded protein in its precise shape for the longest time as the temperature rises? Now we all know that the strongest bond here, by the way, it's asking for the most stable or the strongest bond that can withstand temperature changes. Therefore, it's definitely going to be a covalent bond. And the only covalent bond we have here is the disulfide bond. Covalent bonds are formed by the sharing of electrons, therefore they are the strongest. Now, also peptide bonds are covalent bonds, therefore they are the last to break when temperature rises, therefore the answer is going to be A. Number 12. Protease enzymes are found in many locations inside and outside these cells. 4 of these locations are listed. Which of these locations are sites of intracellular hydrolysis? So intracellular is anything that happens inside the cell. Therefore, it's definitely going to be lysosomes because lysosomes are organelles found inside these cells. Also, let's look at 2, lumen of the stomach. As we all know that enzymes are released into an epithelium. If it's released into an epithelium or any type of epithelium, therefore it's an extracellular, not an intracellular. It's released outside the cell. 3, at a telophase spindle. Therefore this is incorrect. 3, at a telophase spindle. As we all know, at telophase, what happens is that the cell, the 2 sister chromatids move apart and a nucleus forms around both of them. Therefore, this happens inside the cell, and this is correct. For 4, mucus in the trachea. As we said, that mucus is released into an epithelium, therefore it's extracellular, and this is incorrect, and the answer is going to be 1 and 3, which is C. Number 13, an enzyme is modified for industrial use. It has a lower Michaelis-Menten constant, Km, than the unmodified enzyme. What is true of the modified enzyme? So the data that we have here is a lower Michaelis-Menten constant. Now, what is a Michaelis-Menten constant? It's basically a measure of affinity of the enzyme to its substrate. Therefore, the higher the Km, the lower the affinity. For those who don't know, affinity is the attraction it has towards the substrate, the attraction the enzyme has towards its substrate. So the higher the affinity, the more collisions occurs with the substrate. Now, the lower Km, the higher the affinity. Therefore, they are inversely proportional. And here it says it has a lower Michaelis-Menten constant, therefore the affinity has increased. Therefore, the only correct answer must be B, because Michaelis-Menten constant only specifies the affinity. Number 14, a decrease in some factors will increase the fluidity of the cell surface membrane. So decrease increases the fluidity. Which pair of factors, when decreased, will increase the fluidity of the cell surface membrane? Now let's look at the suggestions that we have here. For A, distance between phospholipid molecules decrease. Now, what happens when the distance between them decrease? This means that phospholipid molecules pack closer together. Actually, when they pack closer together, the membrane becomes less fluid. It does not increase the fluidity, it actually makes it less fluid because now they are packing way closer together. So this is incorrect. And B, distance between phospholipid molecules. Now, distance between phospholipid molecules, we just said it's incorrect and we mentioned the reason. And here we have temperature. So for temperature, we all know when decreased, the fluidity also decreases because the kinetic energy decreases. Therefore, this is incorrect too. Now, for C, proportion of phospholipids with saturated fatty acids chains. Now, phospholipids with saturated fatty acid chains, when they decrease, they actually increase the fluidity because unsaturated fatty acids increase the fluidity. Why, you would ask? Because unsaturated, because there are kinks in the chain due to the double bonds which cause the kinks. Therefore, the phospholipid molecules pack more loosely. If they pack loosely, this means that it's more fluid. Therefore, this is correct. Now, proportion of long fatty acids chains, there's a decrease in it. This is correct, it will increase the fluidity. The reason for this is, as you know, the long fatty acid chains, this means that there are more bond interactions, stabilizing the phospholipid bilayer. Now, if there's a decrease in these long fatty acids, means that the bonds stabilizing them decrease. Therefore, it becomes more fluid. Therefore, C is going to be the correct answer. Question number 15. The diagram shows a cell surface membrane. Which diagrams about the labeled molecules in the membrane are correct? 1 is involved in the diffusion of ions. As we could see, this is a phospholipid molecule. And as we all know that phospholipid molecules only allow the diffusion of nonpolar or hydrophobic molecules, because the phospholipid or the fatty acid part is actually nonpolar. Now, what is involved in the diffusion of ions are actually either carrier proteins or channel proteins or both. So there must be a membrane protein involved in the diffusion of ions specifically. Now for 2 is involved in facilitated diffusion. Now, as you just mentioned, because this is a membrane protein, then it's definitely involved also in facilitated diffusion. Facilitated diffusion is the diffusion of substances down concentration gradient through a transport protein. Therefore, this is correct. 3 is involved in the recognition of antigens. As we could see here, this is a glycoprotein bound to a membrane protein. Therefore, this could act as a receptor, triggering a second messenger or cyclic AMP, or activates a G protein. Therefore, we could just leave it for now. HER4 is involved in the membrane fluidity. So this is a cholesterol molecule. And we all know that if the temperature increases, the cholesterol molecules lowers the fluidity, and if the temperature decreases, the cholesterol molecules actually increase the fluidity. Therefore, this is correct. Now, coming back to 3, as we said, it acts as a receptor, and because we said that all ones are wrong, therefore D must be the correct answer. Question number 16, equal-sized potato pieces were placed into test tubes containing equal volumes of different concentrations of sucrose solution and left for 30 minutes. All other variables were controlled. After 30 minutes, the potato piece in one of the concentrations of sucrose solution had not changed in size. What can be concluded from this result? Now, it hasn't changed in size. This means that their water potentials are approximately the same. So, similar water potentials. Therefore, there has not been any water movement either in or out. Now, 1, there's no net movement of water into or out of the potato. We just mentioned that because they haven't changed size. Now for 2, the water potential of the potato is the same as the water potential of the sucrose solution. We have just mentioned that. This means that there has not been a net movement of water. Therefore, it is correct for 3. The concentration of sucrose in the potato is the same as the concentration of sucrose solution. Now, this was a very common misconception. This is actually incorrect because we could say the concentration of solutes, not sucrose. We cannot guarantee that all that is inside the potato is going to be the sucrose. It might also have other solutes involved, such as ions, for example. Therefore, we cannot guarantee that both of them are going to have the same concentration of sucrose specifically. They might have the same solute concentration, but not necessarily of sucrose. Therefore, 3 is incorrect, and the answer is going to be B. 19. Which features of stem cells enables them to replace cells in tissues such as skin? A. There are undifferentiated cells that are present at birth. Okay, this is correct, but it does not give a reason why it allows them to replace cells in the skin. Therefore, this is incorrect. B. They differentiate to form skin cells. Beware, this was also a common misconception. The stem cells themselves do not differentiate, because if that was the case, then we would definitely run out of stem cells, because if all of them differentiates, we're going to run out. Therefore, this is incorrect. C. They divide by mitosis to supply some cells that can differentiate. This is totally correct, because what happens? So, a stem cell here divides both into another stem cell, then a differentiated cell, or a cell that can differentiate. Therefore, we have a constant supply of stem cells. Otherwise, for B, we would definitely run out of stem cells. Therefore, C is the correct answer. Question number 18. During prophase, a scientist stains the chromosomes of a diploid animal cell with a fluorescent dye to stain telomeres. This cell has 32 chromosomes. How many telomeres will the scientist observe? Now, the first hint here, it says during prophase. During prophase, a chromosome looks like this and has 2 chromatids. So, this is a chromatid. Now, telophase and anaphase, the chromosome has a single chromatid, because each pair of sister chromatids have moved to the opposite side of the cell. Now be aware of this point. Now here it says how many telomeres. Now as we all know, there are 2 telomeres at the end of each chromatid. And if we know that per chromosome we have 2 chromatids, therefore we're going to have 4 telomeres per chromosome. 2 at the end of each chromatid. Therefore here, because it says there are 32 chromosomes, there we will multiply 32 by 4. This gives us the question number 19. The diagram shows the cell cycle. During which phase do chromosomes condense and become visible? So the only phase which chromosomes condense to stop protein synthesis is during mitosis. Now mitosis happens in the phase right before cytokinesis. Therefore D is going to be the correct answer, being mitosis. This is the only time which the chromosomes condense, and it's during prophase specifically. Question number 20. What is correct for thymine? Now thymine is a pyrimidine, and cytosine and thymine are pyrimidines. Pyrimidines are single ringed structures, so they have a single ring. Now let's see the suggestions that we have here. So has a single ring structure. Yes, this is correct because it's a pyrimidine, and it is a pyrimidine. This is correct. Joins is complementary base with 3 hydrogen bonds. As we all know, what joins with 3 hydrogen bonds are going to be cytosine with guanine is what joins with 3 hydrogen bonds, and thymine joins with adenine with 2 hydrogen bonds, so they join with 2. Therefore this is incorrect, and the answer is going to be B. Question number 21. A short piece of DNA 15 base pairs long were analyzed to find the number of nucleotide bases in each of the polynucleotide strands. Some of the results are shown below. Now here it says 15 base pairs long. This means that each strand has 15 bases. Just to make things clear, and we know that strands bind together by complementary base pairing. Now here we have thymine. In strand number 1 we have 3. It binds complementarily with adenine. Therefore, if there are 3 thymine in strand number 1, then in strand number 2 there must be also 3 adenine. Same with thymine in strand number 2. If thymine in strand number 2 has 4 bases, therefore also adenine in strand number 1 is also going to have 4 bases. Now here we have cytosine in strand number 1. It binds to guanine via complementary base pairing. So if strand number 1 has 6 cytosine, then in strand number 2 is going to have also 6 guanine. Therefore here it's asking how many nucleotides containing guanine were present in strand number 1. As we all know, here we have 15 bases in each strand. Therefore we add up 4 plus 6 plus 3, and then we subtract it from 15, which gives us 2. Therefore the answer is going to be A. Scientists grew bacteria in a medium containing heavy nitrogen N15 as the only source of nitrogen. After many generations, all of the bacterial DNA contained heavy nitrogen of N15. These bacteria were then moved to a heavy nitrogen medium into a medium with only light nitrogen N14. Some bacteria were collected from each of the next 3 generations. This means that it has been left to divide 3 times. This is very important. And their DNA was analyzed. Hybrid DNA contains both heavy and light nitrogen, which shows the correct DNA of the first and third generations. So here we want to see the first division and the third division. Now, how we do this? Ideally, the values must be memorized just to make it easier for the students, but I'm going to explain why we have these values. Now, let's start with N15 and N15. Now it has now divided. It's the first generation. Divided, this means that N14 has been added to it. Now adding N14, this means that now we have N14, N15, and N14, N15. This is the first generation. Now we can clearly see that it's all hybrid. It's a mix of N14 and N15. Therefore, all hybrid is correct for the first generation. Now let's do the second generation. We're adding more N14, so N14, N14, N15, N14, N14 and N14, N15 and N14. Now we have here half light nitrogen, so 50% light, and 50% hybrid. This is for the second generation. Now for the third generation, let's see what we've got here. This is for the second and for the third. Now we are adding N14, more N14. Now what we have here is that we have a total of 8. So here we could see that we have almost 2 out of 8, which is this one and this one, hybrid. Therefore, we have 25% hybrid, so quarter hybrid. And hybrid is N15, N14, and the rest, so 75%, is going to be the light, because there are 6 out of 8 which are all light, which is N14 and N14. Therefore here 1/4 is hybrid and 3/4 of light. Therefore the correct answer is going to be B. I hope I made the explanation clear, but ideally you must memorize all the values up to until the third generation. So this is the third generation. Question number 23. A population of bacteria is exposed to the antibiotic penicillin. Most of the bacteria die. However, some bacteria in the population have an allele coding for an enzyme that breaks down penicillin. These bacteria are able to survive. So penicillin basically prevents the growth of bacterial cell walls because it inhibits the enzyme that forms cross links between bacterial cell walls. Now, which could explain how these bacterial cells acquire this allele? 1. An error during DNA replication. Basically, this means that a mutation took place. Now, this is correct. The only time an allele, so basically a new gene, is formed is during a mutation. 2. An error during transcription. This is incorrect. An error during transcription would most likely produce a faulty mRNA molecule, but it does not explain how it acquired an allele. So the gene hasn't changed itself, it's just the mRNA molecule that has been altered. So this is incorrect. 3. An error during translation. So if an error during translation took place, then there will be a faulty polypeptide chain, but this also does not mean that a new allele would be acquired. So a totally new gene would be acquired. Therefore, the only correct answer is going to be C. Question number 24. A dicotyledonous leaf has a palisade mesophyll layer that's approximately twice as thick as the spongy mesophyll layer. Which plant diagram is correct? Now, first of all, in a cross-section through the leaf, the xylem is always above the phloem. Therefore, this is correct. So the xylem is always going to be above the phloem. So all these diagrams are correct, which shows the phloem over the xylem. Now, here it says that the palisade mesophyll layer is approximately twice as thick. Palisade mesophyll layer is the upper layer, so it's going to be thicker than the spongy mesophyll, which is at the bottom. If it's going to be thicker, then it would take more space. Therefore, C is going to be the correct answer. 25. A number of processes contribute to maintaining a water potential gradient in plants, allowing water to reach the highest parts of a plant. Which processes are responsible for maintaining this water potential gradient? So, water potential gradient are maintained by means of transportation of water through the plant. So that's what we're looking for. Now, One, capillarity. Capillarity is basically another word for adhesion. And adhesion is how water is transported, how water molecules is transported by the hydrogen bonding between hydrophilic cellulose and water molecules. This helps and aids in the transportation of water up the stem. Now, so this is correct. 2, osmosis. Osmosis is the movement of water down water potential gradient. So this also aids for the transportation of water and helps maintain water potential gradient across plant cells. Therefore, this is correct. 3, transpiration. Transpiration is the loss of water through the stomata, loss of water vapor through the stomata. Therefore, it pulls more water molecules down the root because it forms a water potential gradient. Therefore, more water is pulled upwards to the plant. Therefore, transpiration is correct and A is going to be the correct answer. Number 26, what is a correct statement about the movement of substances absorbed by roots? A, ions have to pass through cell walls to cross the endodermis. Now, as we all know that endodermis have a Casparian strip. Casparian strip inhibits the apoplast pathway, and apoplast pathway is the movement through cell walls and only allows the symplast pathway. Therefore, ions cannot possibly move through cell walls to cross the endodermis while there is a Casparian strip which inhibits the apoplast. And as we mentioned, apoplast is the movement of water through cell walls. Therefore, A is incorrect. B, ions move through the symplastic pathway only until they reach the Casparian strip. Again, ions move both through the symplast and apoplast until they reach the Casparian strip, but here it says symplastic pathway only until they reach the Casparian strip. Actually, until it reached the Casparian strip, it can only cross by symplast. Therefore, it cannot cross by apoplast. So it is incorrect. C, soil water has a more negative water potential than the xylem sap. Now, more negative water potential basically means a lower water potential. This is not possible because if soil has a lower water potential than the root, then how possibly would water move by osmosis down water potential gradient into the root? This is not possible. Then the soil water must have a less negative water potential or a higher water potential to be able to move into the roots. Therefore, this is incorrect. The water passes through the symplastic pathway along a water potential gradient. This is correct. Water passes through a symplast pathway down water potential gradient. A symplast pathway is the pathway crossing cell walls into the actual cell itself, into the vacuoles. Therefore, D is going to be the correct answer. Question number 27. Which arrangement of 4 molecules of water shows how water may cohere when moving up xylem vessel? Now, as we all know, when water moves up the xylem vessel, they move up in a continuous column, each water molecule pulling the one under it upwards. Therefore, the only correct answer in this case must be B, because here it's showing a continuous column, each water molecule pulling the one behind. It cannot possibly be A, because here it's showing that they are in a circle. This does not actually happen. And for C, here it's assuming that hydrogen atom forms 2 hydrogen bonds with 2 oxygen atoms. This is not possible. An oxygen atom is what forms 2 hydrogen bonds with hydrogen atoms, not the opposite. Same for D. Here it's assuming that also hydrogen atoms forms 2 hydrogen bonds with oxygen atoms. Therefore, this is incorrect here. Therefore, the only correct answer must be B. Question number 28. Which statement about sucrose loading into companion cells and then into phloem sieve tube elements is not correct? So let's quickly outline the process of loading. Now what happens? Let's assume that this is the companion cell and this is the phloem sieve tube element. Now what happens is that a companion cell pumps hydrogen ions to its cell wall. So against concentration gradients by proton pumps. So the first step is proton pumps, and this is an active process that hydrogen ions are pumped out. Now what happens is that adjacent mesophyll cells also form sucrose. So there's sucrose on the outside. What happens is both a hydrogen ion and a sucrose move back into the companion cell. Okay, for hydrogen being down concentration gradient and sucrose against concentration gradient into the companion cell. Now, once the sucrose is inside the companion cell, it moves by normal diffusion into phloem sieve tube elements. Now, let's see the suggestions that we have here. A. Hydrogen ions and sucrose molecules move into the companion cells using a carrier protein. This is correct because a carrier protein is the co-transporter protein. We just said that both of them move into the companion cell by a protein called a co-transporter protein. Hydrogen ions are pumped out of the companion cells by active transport. Now, this is correct. We said that this is an active process and hydrogen ions are pumped outside the companion cells by active transport by proton pumps. And by the way, here the question is asking about which one is not correct. C. Sucrose molecules are carried into the companion cells down the concentration gradient for sucrose. So, sucrose molecules are actually carried against concentration gradient. The reason for this is because we said that adjacent mesophyll cells synthesize sucrose, and therefore there's a higher concentration of sucrose outside the companion cell, and they moved into the companion cell through that co-transporter protein against concentration gradient, so not down the concentration gradient. This is incorrect. Therefore, this is the incorrect suggestion, and the answer is going to be C. Sucrose molecules move from a companion cell into a sieve tube of the phloem through plasmodesmata. Obviously, this is correct because sucrose moves by diffusion from the companion cell into the phloem sieve tube elements through the plasmodesmata because it allows the transport of substances between plant cells. Therefore, the only incorrect answer is going to be C. Question number 29. What explains why the left and right sides of the heart contract simultaneously? So let's just briefly draw the diagram of the heart. Contract simultaneously is basically contract together. By means both atriums, the right and left, contract together, then both ventricles, also right and left, contract at the same time. Now A. Both atria have a sinoatrial node. This is incorrect. The sinoatrial node is only found in the right atrium. And here is the right and left. It's only found in the right atrium. B. Both sides of the heart are supplied by the same coronary artery. Coronary artery has nothing to do with contraction stimulation. It has something to do with the sinoatrial node, not with the coronary arteries. Purkinje tissues are found at the bases of ventricles, here and here. Therefore, they never link the two sides of the heart, and this is incorrect. D. There's no barrier to electrical excitation between two sides of the heart. Yes, this is correct because there is no barrier. This means that the excitation waves from the sinoatrial node in the right atrium could also pass to the left. And the excitation waves from the AVN node could also pass to both ventricles, both left and right. Therefore, this is going to be the correct answer. Question number 30. The statements list some of the events in the cardiac cycle. They are not in correct order. Which statements describe the third of these events to occur in the cardiac cycle? Now, the cardiac cycle starts by the sinoatrial node in the right atrium initiating an excitation wave. So it initiates an excitation wave in the right atrium. So, therefore, this one is going to be the first step. Now, what happens is that wave of excitation spreads across the atrium. Therefore, this is the second option that we have here. And then what happens after the spread? The atria contract. Therefore, the third to occur in these events of the cardiac cycle is going to be D, which is 7. Question number 31. The diagram shows pressure changes in different parts of the heart during one cardiac cycle, during which period are the semilunar valves open and the atrioventricular valves closed. So an easy way to interpret this type of graph, which you would normally get in any past paper, is through a mnemonic called COCO. So close, open, close, open. This must be memorized. Now let's see here what we're looking at. We're looking at aortic pressure. So aortic pressure has something to do with the semilunar valve, because as the aortic pressure increases, this means that the semilunar valve opens. So this line here has something to do with the semilunar valve. And then we have ventricle pressure. This line here represents the ventricle pressure. Now, ventricle pressure also has something to do with the semilunar valve, because when it contracts, the semilunar valve opens to allow blood into the atrium. Now, for the last line here, we have atrial pressure. Now, atrial pressure, so this is semilunar, and atrial pressure has something to do with the atrioventricular valve. So atrioventricular valve, because when the atria contract or the pressure on the atria increases, this means that the atrioventricular valve are pushed open. So that's what we're looking at here, during which period are the semilunar valves open and the atrioventricular valves closed. So the semilunar valves are going to be open when the ventricular pressure increases. So in this case, it's going to be C, because here is the most increase in pressure of the ventricular pressure. And now, the atrioventricular valves are closed also during C. The reason for this is aortic pressure is the lowest in this period. And by the way, do not be confused and do this answer as B, because here it's asking about the period, which they are open, not starting to open or starting to close. There's a very huge difference in the terminology used here, so beware, and the answer is going to be C. Question number 32. Which diagram shows the events that occur during transport of carbon dioxide by the blood? Now let's start with A. Now, it might look to you as a normal diagram, but here there's a huge error. Here it's assuming that water followed by the enzyme carbonic anhydrase forms carbon dioxide and hemoglobin. Now, this is incorrect. For B, here we can see that carbonic acid has dissociated into hydrogen ions and bicarbonate ions. But here there's a huge error, that bicarbonate ions leave the red blood cell into the plasma. They never stay in the red blood cell. And the hydrogen ions, some of it leave the red blood cell and some of it stay inside. But here there's a huge error to do with the bicarbonate ions, therefore this is incorrect. For C, here we can see a completely normal diagram, which is correct, because carbon dioxide plus hemoglobin forms carbaminohemoglobin, and carbon dioxide plus water, along with the enzyme carbonic anhydrase, forms carbonic acid. This is also correct, and it dissociates outside the red blood cell as we just mentioned, so C is going to be the correct answer. Now D. The error here in D is that here it's assuming that carbonic acid has left the red blood cell first, then started dissociating. This is incorrect. Carbonic acid dissociates inside the red blood cell, then bicarbonate ions leave the red blood cell. Therefore, this is incorrect, and the correct answer is going to be C. Question number 33. Which structures are found in bronchi? Now, this is mainly to be memorized. Here we have smooth muscle. This is correct. Cartilage. For bronchi, we have irregular cartilage. But for trachea, we have C-shaped cartilage. So, cartilage is correct. And now finally we have goblet cells. This is correct. Bronchi also have goblet cells. Both of these three structures must be memorized as part of the bronchi, and the answer is going to be B. Number 34, which tissue in the respiratory system is correctly linked to its function? Now let's start with A. Cartilage. Stretch and recoil to force air out. So what stretches and recoil are the elastic fibers? So this is incorrect for B. Ciliated epithelium gives protection from suspended particles in the air. This is correct. The reason for this is that ciliated epithelium has goblet cells, which in turn releases mucus, which traps pathogens, and then the cilia on this ciliated epithelium could sweep that mucus to the back of the throat. So this is correct. For C, elastic fibers contract and relax to adjust the diameter of bronchioles. This is actually smooth muscle is what contracts and relaxes. Elastic fibers stretch, they don't contract and relax. So this is incorrect. The smooth muscle keeps trachea and bronchi open. This is incorrect. It's actually cartilage. Therefore, the correct answer is going to be B. Number 36, the diagram shows some of the pathogens that cause disease in humans and some of the ways they are transmitted. Which row matches the correct number for the pathogen with the correct letter for their mode of transmission for cholera and measles? So let's start with cholera. So the pathogen causing cholera is Vibrio cholerae, and it's a bacterium. So one is correct, and it's transmitted by fecal oral route or water contaminated with feces and sewage. Therefore, X is going to be correct for cholera. For measles, we know it's a virus and it's transmitted by airborne droplets through coughs and sneezes. So 3, virus, and Y, coughs and sneezes, is going to be correct, and the answer is going to be A. Question number 37, which factors would increase the global distribution of malaria? So 1 is incorrect, a fall in annual rainfall. The reason for this is that here it says increase the global distribution of malaria. As we all know, that larva grows in the ponds produced by rainfall. So as the number of ponds increases, or the size of them increases, the more malaria or Anopheles mosquitoes are able to reproduce, and therefore malaria spreads. Therefore, here we are limiting the number of insect vector, which is female Anopheles. Therefore, this would actually decrease the global distribution, and it's incorrect. 2, an increase in the use of antibiotics. An increase in the use of antibiotics actually decrease the global distribution. So this is incorrect. 3, a rise in global air temperature. Okay, a rise in temperature, usually female Anopheles or mosquitoes favor an increase in temperature. So a rise in temperature would mean that it would increase. So this is correct. 4, increasing irrigation of land for farming. So increasing irrigation means that there is going to be more water ponds available. Therefore, malaria or female Anopheles are able to spread and increase. Therefore, the correct answer is going to be D. Question number 38. The antibiotic was discovered in Jan, and it kills bacteria such as staph and mycobacterium. Most of the antibiotics work by binding to proteins. This antibiotic binds to lipids that are used in the synthesis of bacterial cell walls. This means that it's unlikely that bacteria will quickly develop resistance to that antibiotic. Which statements explain why bacteria are unlikely to quickly develop resistance to this antibiotic? Now, as we all know, a resistance happens when there's another allele obtained, or a mutation, or a change in the gene. Now, here it says it binds to lipids. If you don't know, lipids are not coded by genes. What is coded by genes are polypeptide chains. So, a mutation or change in genes does not mean that it would form resistance to lipids also, because I've just made it clear that lipids are not coded for by genes at all. They are formed by metabolic pathways. Now, a mutation in the gene coding for a protein allows selection for resistance. I just mentioned that this is incorrect. 2, the antibiotic binds to a lipid rather than to a protein. Therefore, this is correct. 3, the structure of a lipid is not encoded by the DNA. We just mentioned this is correct, and the answer is going to be D. Question number 39. Which row correctly describes the type of immunity gained from being injected with an antitoxin? So an antitoxin is basically antibodies. Be careful, antibodies, not antigens. Antibodies do not stimulate an immune response or the production of memory cells. Now, because it's injected with antibodies, this means that it is not active, it's passive, and it's artificial because it's being injected. It's not breastfeeding, and it's passive, and it's not natural. So C is going to be the correct answer. Question number 40. Where are antibiotics found during an immune response? Now, during an immune response, B cells are stimulated to divide, the ones which are complementary to that antigen, forming plasma cells. These plasma cells release antibodies. Therefore, these antibodies, what happens is they bind to the surface of the pathogens, surface receptors of pathogens, and because they are released by plasma cells, they are going to be found in the bloodstream, also in the blood, in the plasma. Therefore, A is going to be the correct answer. I hope you found this video useful, and if you feel that it gave you any value, I would highly appreciate it if you would subscribe to this channel and like this video.